Okay, we might have troubles with that. It seems lately I've been having troubles with connecting and disconnecting, so that's why we're having separate files to watch here. But anyway, as I was saying, in the prokaryotic and the eukaryotic flagella, what we're seeing is another example of convergent evolution, right? Different structure, uh, uh, but similar function, right? So these would not be examples of homologous structures. They would be examples of analogous structures, aren't they? Different structure, same function. Swimming in the dolphin using its flipper to swim, the fish using its fin. All right. So if they have the ability to move, then let's talk about that for a second. Taxis is the word we use to refer to organisms, whether they're prokaryotes or not, moving to or away from something. So we can call it positive or negative taxis, right, or taxa. And you can be, and that thing you're moving towards or away from can be chemicals. Like, for example, there are many bacteria that have sensors on the surface to sense environmental chemicals, nutrients, that sort of stuff. Photo light, especially if you're a photosynthetic bacterium organism, and you live in a lake during when the sun comes up, you want to be able to respond to light. So you have phototaxis. Some are oriented themselves to the earth, magnetic fields. We call it geotaxis. Organisms that perhaps would want to know where the nutrient-rich lake bottom would be. The geotaxis ability would be able to tell them, in a very broad sense where the center of the earth is and which part is further away from the center of the earth. You know, if you're swimming down into the depths of a lake, you're getting closer to the center of the earth, right? So that's how they use that. Although eukaryotic cells are quite a bit of organization, prokaryotic cells are still organized. Now, no nucleus, right? These are anuclear. They have a nucleoid region. They have no internal compartmentalization. You remember that was one of the big things that separated prokaryotic and eukaryotic, right? Eukaryotic cells, each of the organelles inside of eukaryotic cells has a membrane around it. Remember, the mitochondria and the chloroplast had double membranes, which is why we think they were free living at one time, because that was their membrane they brought along. And then in eukaryotic cells, every organelle gets its own membrane, right? So if you have a membrane and then another one comes along with a membrane. Hi, Thalia. Nice, somebody joined me. Excellent, cool. Yeah, I started a little bit earlier, but hang on, and we'll keep going. Good. Another human to talk to. But feel like I've just been talking into space for a while. So as I was saying, prokaryotic cells, yeah, they don't have a nucleus like you learned in Bio 1, and again, no internal membranes around the organelles, but they do have a lot of infolding. See, over here in the right-hand side, all of these respiratory membranes and thylakoid membranes and this aerobic and this photosynthetic prokaryote are the result of infolding, like folding in on themselves. And if you remember, if you go back to respiration and you go back and you look at photosynthesis, all of that was going on, or much of that was going on across the membrane, wasn't it? You remember in respiration, you were pumping protons across the membrane? And then they flowed back through ATP synthase. You did the same thing in photosynthesis too, right? In photosynthesis, you were pumping protons across the membrane. It's all about membranes. You remember diffusion and the protons flowing back through is Mother Nature's natural tendency to balance things out. Living things take advantage of that. But now I'm going to say something that sounds pretty obvious, but think about it for a second. You can't have more of something on one side than the other unless there's a side. So I can't pump protons across something unless there's a membrane. That's why cellular membranes are so important. So in aerobic prokaryotes, they don't have a mitochondria to do respiration. Photosynthetic prokaryotes don't have chloroplasts, but they do have their membranes. So they do the same kind of process, but they don't have a separate organelle. Very important, because a very big leap in evolutionary biology as you move from prokaryotic to eukaryotic. A genome is the collective set of genetic information in a species. So you yourself have a genome. That's the collection of all your genetic information. If we were sitting in class, I could wave my hands around the room and say, all of us represent a genome. all of us on Lake Sumter campus tomorrow morning would would represent a genome. You get the idea, right? In general, prokaryotes have a relatively simple genome. In other words, they don't have nearly as much DNA as we do, although they don't have introns. Remember, prokaryotic DNA does not have introns. Introns were the intervals, the part in your DNA which are not read. Remember, you have the introns and the exons. It always seems to be backwards to me, but exons are expressed, introns are the intervals. There are no introns in prokaryotic DNA. It's clean all the way through. It's all usable stuff. They also have another piece of DNA, an accessory loop of DNA, maybe with a few genes, called a plasmid. And these plasmids are interesting because what they typically do is they impart special features or special characteristics like resistance to antibiotic, special contingencies, different food stuff, different things they can extract energy from. Now, these plasmids can replicate themselves, make copies of themselves independent of the main genome. In other words, well, you can see in the picture, this bacterial cell on the right, they've taken its chromosome and exploded it out. This is all its chromosome. All of this winding here is its one singular chromosome in one big loop. And then the plasmids, here are some plasmids. So those can replicate, and those are DNA too. And as I said, they can actually code for certain special contingencies like antibiotic resistance. And that's one of the problems we have with bacterial infections and resistance to antibiotics, because if I have a plasmid, if I have an accessory piece of DNA that makes me immune to an antibiotic, I can pass it to you through that 6-pili, that picture I was showing you the previous slides ago, that conduit. I can pass you that plasmid. And so you don't have to hope a mutation comes along or reproduction gives you that characteristic to be able to deal with that environmental condition, right, the antibiotic. You actually get the genetic information from another organism. Kind of spooky, huh? It means these guys can vary, they can evolve and change very quickly. Right? They don't have to rely on necessarily coming up with their own mutations. If they get a mutation that's working in their plasmid, like, oh, now, you know, I can now digest this kind of fungus that grows in, you know, the floor of the bathrooms, you know, that kind of stuff, then I can pass that to other members of my group and we can all do it. Genetic variability. That's an interesting question because, as I said, these are all asexual reproducers, okay? So mutations can arise spontaneously, just like they do in humans. They can arise in bacteria the same way. Radiation and chemicals can encourage that or speed that up. Mutations in plasmids we just talked about can spread very rapidly in a population. MRSA, methicillin, which is an antibiotic-resistant Staphylococcus aureus. Staphylococcus would then be round, but shaped like grapes, right, the staphyla. But MRSA, the Staphylococcus aureus, became resistant to methicillin because we believe of passing of plasmids. So a group of those Staph aureus, through mutation, through luck, came up with that plasmid that made them resistant to methicillin, and then they passed it to all of their other Staphylococcus aureus. And you get a whole group of them. Now, this is, again, what we call horizontal gene transfer. Vertical gene transfer is I pass the genes to my offspring or I got them from my parents. Horizontal is you and I exchange genetic information. Right? That's what those 6-pili are for. They're exchanging genetic information. You might be exchanging a plasmid through there, for example. Okay, other ways in which we can, or other ways prokaryotes can generate genetic variability is through something called transformation, or conjugation, or something called transduction. Now, I'm going to get into each one of those in just a second, but I want to go back and remind you that, as we said, most mutations in evolution, most mutations to DNA are detrimental, right? Most mutations cause the organism to be less fit, so it doesn't survive. So we said that among sexually reproducing organisms like you and I, mutations are not a great source of variability because very often they don't end up well. Or if they do, they end up occurring in the exons, the part that is, or I'm sorry, the introns, the part that's not read anyway. How do mutations get away, or how do bacteria get away with using mutation as a source of variation? That's one of the common sources of variation in these organisms. It's just the mutations that we have too. How do they get away with the fact that most of the time mutations are bad? Reproductive rates, right? These organisms have incredibly high reproductive rates. So therefore, they can deal with, 1 out of a million mutations being beneficial because they're making, I don't know, hundreds of millions of offspring, right? So it's a numbers game once again. If you play the numbers, then you can go ahead and these organisms can actually get away with using mutations. All right, but transformation is absorption or integration of fragments of the DNA from the environment, right? So sometimes what will happen is an organism dies. Your DNA doesn't necessarily disintegrate after you die, but the DNA is in the environment, and a bacterial or prokaryotic organism come along and actually pick up that piece of DNA, right? That's what the Griffith, by the way, back in Bio 1, when you learned about the mouse, you remember the Streptococcus that was studied by Griffith. There was the R and the S form, and one was virulent, one was not. If you don't remember that, go back and take a look on that. But what was happening, the reason why one bacteria got changed into another is through transformation, like the picture you see up above. So the cell dies, releases pieces of the DNA. A part of that is taken up by another cell, sometimes even across species line too, by the way. Sometimes they can pick up DNA from another live cell from a different semi-related species. The DNA that gets incorporated, and guess what? Now we have some genetic variability. Now that occurs naturally. In the lab, we do it artificially. In the lab, we actually program bacterial organisms to produce a genetic product, like, for example, insulin, right? You get E. coli, you get a bunch of E. coli, grow them up in a big vat, molecularly clone them so they're all the same, and each one, before you molecularly clone them, or the one you start with, has the gene for making insulin. Now, E. coli have no reason for insulin, right? But they make the insulin as excess, and we use that. It's like we're farming it, if you will. So we can use this ability or this tendency of some bacterial organisms to pick up a piece of the DNA from the environment to actually pick up a piece of DNA that we like, right? Something we can do something with. In conjugation, we're going to use the sex pili, right? So we got transformation, conjugation. So as you see in the picture, there's the plasmid in orange. Notice in the picture too, look closely. When the sex pili, when the conduit is formed and the plasmid is being passed, only one strand of the DNA is being passed, right? All you need is one strand, because you remember Chargaff's rule, as long as you have one strand, you can develop the other one. And it also diminishes the chance of a mistake being made in the DNA being replicated. Remember, that's why your DNA replicates semi-conservatively. Remember that? Here's your DNA. When you replicate, it splits it in the middle, and each side makes a template. And then these come in a bond. Sorry, don't draw so well, but you get the idea. It's tracing. It's like I tell my students, you could draw a better picture of something if you traced a piece of tracing paper. So as that DNA is being reeled off into that recipient cell, we call them F pluses and F minuses. We're going to eventually make both of them F pluses. You're only reeling off half of it. So the half that's going into the new cell, that's being replaced here, the purple, and into the new cell, the purple is being added to the other half it got from the original one. Pretty clever, huh? So this is how they might, these two guys might pass a plasmid, like, for example, the one I was talking about with antibiotic resistance. Now, the last way in which genetic variability that we're going to talk about is used as a vector. And remember, a vector is something that moves something around. Mosquitoes, for example, can be vectors, right? Mosquitoes can move parasitic organisms around. So before I do that, though, I'm going to talk about viruses for just a moment. I don't discuss viruses in this class. There's a chapter on it. More a matter of timing, knowledge. I'm not horribly versed on viruses. Again, if you take a micro class, you'll learn a lot more about viruses in micro. But they're really neat things. They're not really alive. They're not really dead. This is actually a picture of the big green cell being attacked by a bunch of things, type of viruses we call bacteriophages. Bacteriophage. literally means to eat bacteria. This is a bacteriophage right here, this thing. What viruses are is they're a protein, right? All that structures, the legs, the spaceship looking stuff, and then the orange, the same thing. These here, you're just looking at them, you know, from the top, is protein, and then the coil up inside is the DNA. So that's all viruses are. Protein capsule, we call it, or casing, even though it's kind of sophisticated looking, and genetic information. So there's an organization, okay? Viruses do evolve, but they don't really metabolize. They can't, they're not involved in metabolism. All they're doing is they're using host cells to make more viruses. Now, using host cells to make more viruses means there's a chance that bacterial cells that are infected by a bacteriophage can mix up their genetic information that way. So taking a look at this picture here, you see the bacteriophage injects a piece of the DNA. Now, what the bacteriophage is doing is trying to do the same thing that we do with the E. coli and the insulin example I gave you. You see, the phage DNA is information on how to make more bacteriophages. So what it does is it injects it into a donor cell, a prokaryotic cell, right? Causes the DNA to break apart. That phage DNA is then going to be used to create more viruses, more bacteriophages. Okay, now remember, I said a virus is just the protein capsule and the DNA. It doesn't have a ribosome. So it's going to be using the host cell's ribosomes to do that process of protein synthesis. It's going to make some more of those protein capsules. It's then going to start, and literally it's going to cram DNA, and it does it sometimes sloppily, grab big chunks of DNA, shoves it into the head of that virus, and then that virus, that bacteriophage is a type of virus, then goes to another cell, injects it with the phage DNA, which has a little bit of the DNA from the previous cell. All right, so when this part right here occurs, and then we package them back up, some of it gets just bacteria DNA, or I'm sorry, viral DNA, and some of it gets a mixture. Like you get some of the genetics from the original cell. So that's what we mean by a vector, right? The bacteria delivered new genetic information, the virus delivered new genetic information to the recipient cell. This, of course, can be difficult in terms of medical treatment because, of course, now you got viruses and bacteria working together and exchanging genetic information. And if that phage DNA that's being passed around is phage DNA of a virus that causes illness or something we call pathogenic. Pathogenic means something that causes you to become sick. Then, of course, that's problematic from that point of view. Okay, nutrition and metabolism. I know this looks like a pretty complex chart, but only because it has a lot of pictures. If I were to study this for the exam or put it in my notes, what I would do is I would just write the headings and leave off the pictures. I leave the pictures simply for examples. But let's talk about nutrition and metabolism. As I said, these organisms, these prokaryotic organisms can live pretty much anywhere and survive on pretty much anything. But in general, let's kind of break down the way organisms, regardless of what type you are, how they go about obtaining carbon, right? You got to have carbon. You're a carbon-based life form. If you want to get bigger, you're going to need that element carbon to be the foundation, and you need a source of energy. You need somewhere to get energy from to convert, because in order to build things bigger, the laws of thermodynamics, to build those carbon things, right, you're going to need energy. All right, so let's go with the carbon source first, being carbon dioxide. All right, in parentheses, I put the word auto. All right, that's going to give us part of the name. Remember back in Bio 1, we call plants autotrophs because they were self-feeding. Now, the energy source can be different if you're an autotroph. We didn't introduce this in Bio 1, but just to kind of give you a sense of where you were, in Bio 1, when we were referring to plants as autotrophs, we really were saying they were photoautotrophs. The auto part refers to the fact that where they get their carbon is from an inorganic source. Carbon dioxide is inorganic. Remember, to be organic, you have to have carbon and hydrogen. So an autotroph, like plants, use for their source of carbon dioxide, right? They take the carbon dioxide from the atmosphere. They use the energy from the sun to put it together to make food. So this is many plants. These are everywhere, plants. mosses, flowering plants, cone-bearing plants, you know, the things you're familiar with. There are some protozoans that do this, Euglena, for example. Now, if the little blue crossing arrows, if you follow one for the protozoan, indicates that some of these critters in life, protozoans aren't bacteria, of course, they're protozoans, but Euglena actually can feed in more than one way. Euglena can photosynthesize, and Euglena can also feed the way we do, can be a heterotroph. This is kelp, and kelp is not a plant. It's a protozoan, so I put that in the group too. There are some prokaryotes that fit in this group, like blue-green algae, green sulfur bacteria, purple photosynthetic bacteria. They photosynthesize, but they don't use a green-colored pigment. They use a purple-colored pigment. And notice they're also one of the ones that's a double dipper. They can photosynthesize or be a photoautotroph, or they can metabolize in a wholly different way. We'll talk about in just a moment. Now, where you may not understand that you can also be an autotroph but not use sun. You can use the energy from oxidation. Remember, when you oxidize something in chemistry, when you take electrons off of it, you release energy. So you could do some work with that. And if you're oxidizing something inorganic, we would call you a chemo. We would call you a chemo autotroph. All right, so you got a photoautotroph and a chemo autotroph. Sorry there, folks, I confused myself for a second. So chemo autotrophs, and there are those that can oxidize hydrogen, like you see in the first example there. Those are the methanogens. They take carbon dioxide and hydrogen gas, and they turn it into methane, and that releases energy. That's how they survive. Thiomargarita, the sulfur bacteria, they actually can oxidize sulfur or hydrogen sulfide. A lot of these organisms are the ones found down around the geothermal vents because there's a lot of hydrogen sulfide coming out of volcanic activity. Now, light is an energy source. Yeah, light is the energy source. Light's not the building material, right? The carbon. If we're talking about something that's organic in chemistry, we're talking about something with carbon and hydrogen. Yeah. Some of those inorganic oxidizers can use nitrogen, and I don't expect you to know that cycle down below, but these are the nitrifying bacteria. Very important for plant growth, by the way. We'll talk more about that with plants. Now, the other side of the scale is you and I, all right? We're heterotrophs. The hetero part refers to the fact that we get our carbon not from something inorganic like carbon dioxide, but from organic compounds. Now, if you're trying to think, what's he mean? Food. You get energy from food, yes, but food, sugars are organic materials that plants made for us with this photoautotroph, but they're also our source of carbon. So we get the energy off the carbohydrate, and we also use the carbons as our building blocks. So if we are one of those organisms that oxidizes, organic materials, we're a chemoheterotroph. So we oxidize organic materials, okay? We get our carbon from organic materials. We get our energy from oxidizing organic materials, right? The animals, there we are. Fungi, many fungus do it this way. Some plants, this is actually a plant we'll talk about in the plant section that actually makes its living pretty much kind of the way you and I do. Protozoans, Euglena. And this right here is botulism, Clostridium botulism. Clostridium botulism is the disease that you hear a lot about. Botulism kills you. What did I read? 1/10 of an ounce or something in the water supply of New York City could kill everybody in the city. So this is the bacteria everybody freaks out about. But botulism is very, very rare. But it also engages in metabolism the way we do. It uses organic compounds for carbon. It also oxidizes them. Now, if you use carbon as your source of organic compounds, but the energy is from the inorganic oxidation, right? We oxidize organic materials. But if you oxidize inorganic materials, again, back to sulfur, for example, what they do is they oxidize sulfur, and they use the energy from that to then oxidize an organic compound. So it's a little bit different than the sulfur bacteria we saw over here. They oxidize sulfur. These guys, these sulfolobus that you see down here in the picture, they oxidize sulfur. That releases energy. They use that energy to oxidize something else, which is organic. So it's just kind of an intermediate step. Oh, and then one of the neatest ones. Oh, this just blows me away, because if you think about these organisms and their extremophiles, this was a Halomonas titanicea, discovered just recently when Ballard found the Titanic. Titanic is 2 miles down in the ocean, so the pressure, temperature, and there's certainly no light. But they found an organism down there that actually is oxidizing the iron in the iron plates. And when it does that, it's sort of like the sulfolobus. It's basically like one gear turning another. When you oxidize something, you release energy. So you could use that in a chain reaction. You could oxidize sulfur, then oxidize an organic compound and use that to get energy. You could oxidize iron and then use that energy to oxidize an organic compound. And then finally, the purple photosynthetic bacteria are unique in the sense, as a matter of fact, this last process is unique among prokaryotes only. It's called a photoheterotroph. And in this case, you get your carbon sources from other organic compounds, but your energy is not from oxidizing things. It's back like plants do. You use the sunlight. And not a lot of critters out there that way. The more common one, again, is that double dipper, the purple photosynthetic bacterium. So the purple photosynthetic bacterium can either use light as a source of energy and organic compounds, or carbon dioxide as a source of carbon. And if you think about that in a very basic sense, that's that flexibility that prokaryotes have that allowed them to live in so many different environments. Okay, so again, what I would do as far as studying is I'd go through and I'd rewrite this out, maybe in some sort of flowchart the way I have it. I just leave the pictures off, folks. I'd write some of the examples, but if you take away the pictures, this whole chart doesn't look nearly as busy as it does at the beginning. All right, let's see. Yeah, let me keep going. As far as oxygen goes, some prokaryotes are obligate anaerobes, obligate aerobes. In other words, they require oxygen. That would be us. We're an obligate aerobe. Some are obligate anaerobes. Some are actually poisoned by oxygen. Remember, oxygen is a strong oxidizer. All right, oxygen is a strong oxidizer, and that means it destroys things. We just talked about it releases energy, but in the process of releasing that energy, you go from a bigger molecule to a smaller one. You're tearing things down. That's why I always tell my Bio 1 students, just say no to O2. Oxygen's killing you. Every time you breathe, you need oxygen, but it's also destroying your molecules. And some organisms never develop the ability to deal with that. Now, do we deal with it? No, you do eventually die, but at least we can deal with it for a while. These organisms are relegated to just doing fermentation or anaerobic respiration, and then there are those who can switch in between. Now, I know you might be thinking from Bio 1, wait a minute, I remember lactic acid formation. We're talking not in emergency circumstances here, but organisms that can be aerobic or anaerobic depending on where they are. Flexibility. Flexibility is the key here with among these prokaryotic organisms. As I mentioned, in terms of nitrogen fixation, again, we'll talk about this in plants, but the limiting growth nutrient in plants, ecologists and biologists oftentimes refer to what is limiting to this organism. What of all the things it needs is the one it needs the most, okay? And among plants, most plants, the limiting factor in their growth is how much nitrogen there is in the soil. It's why you fertilize crops or fertilize your plants. If you go to a Home Depot and you're buying fertilizer the next time for your grass, you might notice some numbers across the top written sometimes like that, 15, 10, 5. That's the NK concentration, nitrogen, phosphorus, potassium, all right? Now, phosphorus and potassium are two other essential ones, but nitrogen is a limiting factor. Now, the problem is, or I guess the irony of it, the atmosphere is mostly nitrogen. Right? It's 20% oxygen and a bunch of trace gases. It's 75% or so nitrogen. So there's a lot of this atmospheric nitrogen, but plants can't use that nitrogen. In other words, they can't fix it. Over in the red, I've highlighted, or it's highlighted, that's what a lot of these prokaryotic organisms can do. They're beneficial to the plants. They live down in the soil, and what they do is they take in atmospheric nitrogen, convert it through some nasty stuff, ammonia and nitrites, until it becomes nitrates, and that's the fertilizer for plants. Other bacteria actually break down the nitrogenous compounds because that would become toxic. Right, so they're very much involved in this whole thing called the nitrogen cycling. Legumes, bean plants, are very good at fixing nitrogen. The roots of legumes, if you pull up a bean and you looked at the roots, there's all these little lumps all over it, nodules. Those are nodules of colonies of these nitrogen fixing bacteria. So a farmer that wants to naturally make his soil nutrient rich again would plant beans, because they would, obviously the bacteria are fixing the nitrogen for themselves, and they produce a lot of it in the soil for the bean, but there's a lot left over. For cotton farmers used to do this. They plant their field with cotton. Cotton is very bad on nutrients. It strips the nutrients from the soil, and then they would mix in growing beans the following year, because the beans would help put the nitrogen back in the soil. Don't do that anymore, because you can now just put fertilizers on the soil, which isn't any better. But cotton farmers aren't about to grow beans when cotton can bring a lot more money, right? So they don't want to take that year off growing beans. Again, back to this idea of evolution not being rungs on the ladder there, Mr. Aristotle. And keep in mind that we have organisms that engage in what we might call metabolic cooperation. So again, not tissues, sort of multicellular, like Anabaena. Anabaena, says it, a lot of science words are very phonetic. This is Anabaena on the right. Anabaena is filamentous. It's a filament. It's a long string of cells. The ones that are green are involved in carbon fixation, the photosynthetic cells, and they also have nodules like legumes. They have these things called heterocysts, and those involved in nitrogen fixation. So the carbon fixation does the photosynthesis, and the heterocysts provide the fertilizer. One stop shopping. Plaque on your teeth, the things that cause cavities. If you look at it up close, what you basically see, this is your tooth. This is a film, a group, a colony of a bunch of bacteria living together cooperatively. What we mean, again, cooperatively in this sense is all these, and it's Streptococcus again. Streptococcus ferments the sugar in your food, which forms lactic acid. That's what causes the tooth enamel. The fermenting the sugar is to obtain energy. That's why Streptococcus is doing that. And there is more efficiency when they're collected together, when they're clumped together as plaque on your teeth. But again, it's the lactic acid which causes the cavities, right? That erodes your tooth enamel. Oh, diversity, holy cow. My gosh, there's over 10,000 species of prokaryotes. Could be a lot higher, could be 10 times higher. We're not really sure. Again, a lot of the prokaryotes that get attention are the ones we hear about. Some of the early classification schemes involved criteria that we talked about before, kind of what they look like or what they do. And then, of course, recently we've gone to overall looking at the genetics, right? Amino acid sequences, nuclear hybrid, all those things I talked about before. Nothing really new here. This is basically how relatedness, how phylogeny is moving in all groups of organisms. Less of what, look at this one, importance is human pathogens. That's hardly a reason to classify something. That means you would have to put a man-eating tiger and Fifi, your cat, in the same group. And obviously one's a little more deadly than the other. I'll know some nasty house cats. So again, we go to the genetics. I want to point out a couple things. We're going to look at some of these bigger groups of the prokaryotes. These are all the prokaryotes. But I want you to notice something right here. This is the connection between us and them. Take a look at that. That phylogenetic chart says this, at some time a branching event occurred, and off on one branch came the domain, the archaeans, and the other branch became the rest of us, the eukaryotes. I mentioned at the beginning that the archaeans are the more, say, archaic, right? The ones we think represent the earlier organisms on the planet. But again, just to show you that evolution doesn't work in a chain like Aristotle towards perfection, us, right, us eukaryotes, we're more closely related, not, distantly again, but relatively speaking, we're more closely related to those more primitive like prokaryotes, the archaeans, than we are to the other type of bacteria. Interesting, right? Okay, now the next slide, I really only want to focus on the stuff in the red. This is a good little comparison of the three domains of life. First one is the nuclear envelope. I definitely would like you to know which ones do and do not. You should know that, obviously, from Bio 1. We popped it up about peptidoglycan. Now, peptidoglycan, remember we were talking about gram-negative and gram-positive bacteria. Archaeans don't have peptidoglycan. This is another ... link between us and them. The fact that we neither one of us have peptidoglycan. Introns. Bacteria don't have introns, right? It's all expressed DNA, but look at that. We do, and so do the archaeans. Another one of those, maybe. Right? Little bits of evidence. The extremophile part. Bacteria, can they grow at temperatures greater than 100 degrees? Can they grow in temperatures greater than boiling? Not, bacteria cannot. Some archaeans can. We, of course, cannot. Okay, let's look at a few groups of these organisms. Let's go with the archaeans again, the extremophiles. There's the methanogens that I talked about. They're the ones that oxidize carbon dioxide, or excuse me, they oxidize hydrogen. They produce methane. These are the ones in the swamps. If you walk through the swamps and you smell that methane gas that you sometimes smell, those are the methanogens. Methanogens are oxidizing hydrogen. That's where they get the energy from, and they produce methane. Okay, methane is that smelly gas. These are usually obligate anaerobes, so they very often live down in the mud in a swamp and a marsh. People say, where on earth is there no oxygen? Water has oxygen. Yeah, but the mud down below doesn't. That's why sometimes if you're walking through swamp, like a cypress swamp, that smell will come up behind you because you're actually stirring up that gas, that methane gas trapped down in the mud. Methanogens, since they're obligate anaerobes, or I guess I should turn it around, since they don't need oxygen, are good for, very important in herbivore digestion, living in the guts of ruminants, those that choose their cud, cows, right? Because those areas oftentimes become anaerobic. There are those that live in really salty places. They're the halophiles. Halo is salt. These are the ones that you see living around the Yellowstone with all the really pretty colors. My brother-in-law and his my niece and nephew and my sister-in-law went out to Yellowstone. He sent me pictures back, gorgeous pictures of all these different, and those are all different colored bacteria, and those can live with the very salty conditions or very hot conditions too. And the hot ones are what we call the thermophiles. In many cases, the optimum temperature for these guys is 60 to 80 degrees Celsius. So their whole physiology has evolved to live in these really warm climates, ecosystems down around those geothermal vents. Among the bacteria, we have clades. A clade is, we talked about monophyletic groups and clades and all that. A clade is a rough term for a group. It may not have a taxonomic significance to it, but it's a group. We call these, that we're going to look at, the clade, the Proteobacter. So these are all gram-negative. We have the alpha, the beta, and the gamma. Very simply named, A, B, and C, basically. Greek alphabet, it's alpha, beta, and gamma. The alpha proteobacteria very oftentimes have a eukaryotic host, like rhizobium, which you find inside the root cell of a legume. These are one of the nitrogen fixers. Okay? So they live inside the root cell of a legume and help fix nitrogen. They can be parasites or they can be symbionts. Symbionts and parasites, splitting hairs in the difference, but symbiote is an organism that live together. Typically, in symbionts, we're talking about either mutualism, like there's a plus and a plus, right? They both gain a benefit, or one gains a benefit and the other one is neither harmed nor helped. A couple more slides I'll talk about in a little more detail. Beta bacteria, very often found in soil. These are also nitrogen fixers, and Unfortunately, a very common very well-known diplococci beta bacteria is Neisseria gonorrhoeae, which causes the disease gonorrhea, sexually transmitted disease. The gamma are the sulfur bacteria. These are the ones like Thiomargarita that can oxidize sulfur, right? Get the energy from that. And a lot of these are the bad guys, the pathogens. E. coli, which isn't. E. coli is a normal resident in your gut, all right? It's the strain of E. coli. It's kind of like what we would say the breed of E. coli that becomes a problem. The kind that makes you sick is a different strain. Legionella, which is another gamma proteobacteria, Legionnaire's disease. When I graduated high school in the late 70s, a bunch of these people called the Legionnaires had a conference in, it was Philly, and something was in the air conditioning duct and it killed a bunch of them. And we didn't know about it, didn't know about this bacterium until later on. Salmonella is an example of a gamma proteobacteria. Vibrio cholerae, which is another example of a gamma proteobacteria pathogen, causes cholera. Unfortunate disease. You die from cholera from dehydration. Basically, you have diarrhea until you completely dehydrate. Delta and epsilon. So again, we got real fancy with the way these were in the clade. We just call them A, B, C, D, basically. So delta moves along by this gliding motility. It secretes these slimy threads and pulls it along. We used to think a lot of delta bacteria, we used to think that they were fungi at one time, because they kind of look like fungi. This is what they look like. These are the fruiting bodies. It's called a myxobacterium that produces myxospores. You can see, again, the problem with looking at well, you guys haven't looked at fungi too much, but those kind of look like a fungus. But it's not. It's not a fungus. It's actually a prokaryote. Delovibrio is one of those predatory ones. This is Pseudomonas, so this is the prey. There's Delovibrio with a big flagella. Can swim up to 100 microns a second. In scale, that's about 220 miles an hour. Amazing. And then when even better, when it hits, it has to drill through the membrane of the Pseudomonas. And what it does, it has these digestive enzymes, and then it spins like 100 revolutions a second, and then eventually drills into the prey. So it swims up really fast, and then it spins around until it injects itself into the prey and destroys it. That's rough for prokaryotes, huh? If you're someone who suffers from ulcers, there's relief for some of you. Some ulcers we found out recently are caused by actually a bacteria, Heliobacter pylori. You see them there on the right. I guess the good news in that, if you suffer from that type of ulcer, not one that's caused by stress or any of those other things, antibiotic will do the trick, right? So the ulcer can be easily taken care of. The chlamydias, and those are the ones you're thinking about. The chlamydias are a parasitic cell, right? It's dependent upon animal host cells. It's a sexually transmitted disease. It cause blindness, most common cause of blindness worldwide, and most common of the in the United States of the STDs. Look at the chart over here. While the rates of gonorrhea and syphilis have dropped, gonorrhea considerably. Gonorrhea is very simple to treat. Syphilis a little bit harder. Notice the rates of chlamydia have risen. We call it a silent STD. You guys have probably, had training in this in some regard, at least some schooling in this in some regard. A silent STD is a sexually transmitted disease. It's not symptomatic, so you don't know you're spreading it. So you can basically have chlamydia, giving it to other folks without even knowing it. Chlamydia, or a type of chlamydia, Chlamydia trachomatis, it can cause non-gonococcal urethritis. It's an infection of your urethra. Excuse me. The spirochetes, many of those are free living, but treponema, which causes syphilis, is parasitic. This is Leptospira, that's a spirochete. And then this one, Borrelia burgdorferi. Borrelia burgdorferi, this organism here. That's what causes Lyme disease. Lyme disease is from the deer ticks. The tick is the vector, carries the disease around, and then the spiral organism you see in the picture, that's what the bloodsuckers like mosquitoes, ticks, and all that, whenever they take a blood meal, they puncture into your capillary. Well, the first thing your blood wants to do is clot, right? So what those organisms do before they start withdrawing blood, they actually inject a little bit of an anticoagulant into your bloodstream to keep the blood flowing. If they're a vector for a disease, very often when they inject that bit of anticoagulant into you, in there is the parasite. In this particular case, the Borrelia burgdorferi. That spot on the right is that's Lyme disease, folks. They always say it's got the target sort of arrangement to it, the circular targets. We don't have a real problem with Lyme disease here in Florida. We do have deer ticks, and it does show up. But from what my understanding is, it's much more of a problem up near the Midwest. Deer ticks are much more common. Most common kind of tick down here is just the dog tick. Oh, the cyanobacteria, holy cow. We got to talk about these guys. Cyanobacteria are the only prokaryotes who could photosynthesize like eukaryotes, right? So they could photosynthesize in massive quantities. Remember, we talked about how the cyanobacteria changed the earth early on, right? They're the ones that produced all that oxygen. So Anabaena, Oscillatoria, these are the ones that are known for their algal blooms, like Lake Apopka, the green color. What happens in an algal bloom is, in this, again, the word algae can be misleading. There's all kinds of algae. There's prokaryotic algae, protozoan algae, there's even plant algae. So here we're talking about prokaryotic algae. Algal is plural of algae. And what these things will do is they'll bloom. They'll start reproducing like crazy in the lake. And usually that's because a lot of nutrients, usually nitrogen, from fertilizers, it ends up getting into the lake. It's actually called eutrophication when this happens, when a lake starts becoming polluted with nutrients. Not necessarily polluted with waste products, but all those fertilizers dumping into the lake, well, they're good for the obviously for the golf course around the lake, but in the lake, the photosynthesizers, they need that stuff too, so they bloom. And why that's bad for the rest of the organisms in the lake is all these algae start sucking up all the oxygen. So it makes the lake with less oxygen. Fish need oxygen just like any other critter. It also blocks out the sunlight too. So algal blooms, can be a problem. Now, among the gram-positives, we have the colonial ones called the streptomycetes. Or that's the most common name. These are called the actinomycetes. So these are colonial. Again, we get colonial, not multicellular. And then we also have in the gram-positives. Most of I said pathogens are gram-negative, but some gram-positives are worthy of note, like the one that causes anthrax. Oh, there it is. 1 gram can kill 1 million humans of the toxin from Clostridium. And then the staph and the strep infections. If you work in the healthcare industry, you're very familiar with that. Now, not all prokaryotes are bad. There are the good ones too. For example, the ones involved in nitrogen fixation, the decomposers. They're one of the only organisms that can break down this material known as lignin. Lignin is one of the most durable organic materials. It's one of the substances that plants use in their cell walls to stand up. It's a very rigid structure, very complex chemical structure there. And many prokaryotes are the only ones who can do that. Let me briefly go over symbiotic relationships, because we're going to talk about more of those as we go into the different groups. And I've given you a little chart. Symbiotic relationships are two organisms living together, and we classify them based on whether both organisms gain a benefit, so the plus, both organisms are hurt, so the negative, or both organisms, nothing happens, neutral. So in parasitism, species A, which is the parasite, gains a benefit, and species B, which is the host, that's a bad thing. In commensalism, the one organism gets a benefit, and the other one is neither harmed nor helped. The egrets that follow the cows around. The cows kick up the insects. The egrets can feed upon the insects. That's a benefit. The cows, the egret being there doesn't bother the cow, doesn't help the cow. It's a neutral, right? If both of you help each other, like, the clown anemone, the anemone fish and all that stuff, then it's mutualism, which is I'll scratch my back and you scratch your back. Those are pretty obvious. One that might not be so obvious is, why is competition negative and negative? Well, because in competition, nobody wins all of it, right? In competition, even if you get 99% of whatever the resource is, you didn't get 100. So there's always a negative aspect to it. Now, I'll use those terms and those positive negatives and symbiosis in other organisms. So it happens among prokaryotes, but it also happens among the rest of the group that we'll talk about as we go through the semester. As I said, of course, there are good prokaryotes out there. For example, we have a very strong mutualistic relationship with 5 to 1,000 different species of what we call gut microbiota. You know, if you've had digestive issues, the doctor might prescribe Align, which is a probiotic. Not prescribed, you don't have to prescribe, you just buy it. But I found this cool little graphic online to show how many microorganisms are found in different areas of your, like your stomach has anywhere between 100 to 1,000. And then look in your colon, anywhere from 10 to the 9th to 10 to the 12th numbers of microorganisms. Well over 500 different species. Where's E. coli? And they don't list it, but yes, E. coli is a normal part of the lower intestinal gut. So these are the good ones, right? Either the good ones or perhaps they live there, but they don't really harm or help us. Sometimes bacteria are beneficial to us just by being there. If the bacteria is there and it's not harming or helping us, let's just say it's a zero kind of thing in that symbiotic relationship, no, it is taking space and resources. So if it's taking space and resources, it might be preventing a pathogenic bacteria from taking hold, right? So sometimes the benefit is just that you're there. We can use bacterium, especially because they're simple genome, for genetic engineering. You know, engineering or genetics to engineer things. Like, for example, we can produce different kinds of materials like this biodegradable plastic. There's a species of prokaryote that produces stuff called PHA, polyhydroalkanoate, which is a biodegradable plastic. You know, the problems we have with plastic is they don't decompose in the environment. They stay around forever. It takes forever. These things biodegrade. I think they're very sensitive to light. So when they're out in the sun, they biodegrade much quicker. This guy that's spraying stuff all over the rocks, what he's doing is engaging in bioremediation. Same thing they did with the Deepwater Horizon spill out in the Gulf a number of years ago, or summer 2010. There are many bacteria out there that feed upon hydrocarbons, oil. If you think, well, where would they come from? Oil in the ocean, much of the oil in the ocean is natural seepage. It seeps out between the rocks. It's not because we spill things. So, of course, evolution and the environment and oil being energy rich, and organisms, prokaryotes, many of them, or some species of them, have figured out ways to digest the oil. So we turn around and say, okay, when we have an oil spill like the Exxon Valdez or the Gulf oil, we can spray the rocks or dump these bacteria onto the oil in the hopes that they'll eat the oil, they'll consume it. Now they do, but it's a matter of, you know, how much oil was spilled versus how many bacteria actually out there eating it. Like, for example, the Exxon Valdez, which occurred 20 years ago, 25 years ago, if you dig down below the rocks of Prince William Sound, which is where that guy is, there's still oil there. So it looks nice and clean, but the oil's not gone. It's kind of like all the oil in the Gulf. You know, they say, oh, it looks clean now, come to the beach. We threw this dispersant on it. All the dispersant does was chop it up into little tiny pieces so you couldn't see it. And there's even some oceanographers that believe that oil might be these huge layers of oil down lower in the water, deeper in the water, that might be circulating throughout the Gulf. So much as it looks pretty, it doesn't mean it's gone. But bioremediation is something that's exciting because, you know, perhaps we can, I don't know, perhaps we can trick or use Mother Nature to clean up our messes as well. And then there's the bad, right? The pathogens. Look at that. In the early 20th century, 20% of all kids, 1 to 5, died before they were 5. A lot of this had to do with our knowledge of nutrition, you know, prenatal health, but also our knowledge of bacterium. If you keep in mind, a lot of the discoveries in science parallel with technology, right? So as microscopes got better, we got better understandings of different kinds of bacteria, like the one that caused tuberculosis, Mycobacterium tuberculosis. For thousands of years, that was a problem. And we had it whipped, and for the most part, it's under control. But because of our overuse of antibiotics, which basically is, if you think about it, is artificial selection, right? Because even though you treat an antibiotic onto 1 million of these bacteria, not all of them will die, right? Some of them might just be lucky enough to survive. And now we find, especially with the plasmids, there's many out there that are MDR, multi-drug resistant strains. We talked about the exotoxins. The problem with the exotoxins is you can sometimes kill off the organism, but if the toxin is still around, that's what's making you sick. Okay? Well, endotoxins and exotoxins. I'm sorry, exotoxins are secreted, endotoxins are produced by the polysaccharides. Either way, the toxins can still make you sick. Horizontal gene transfer, as I mentioned, were among these pathogens, so we can get virulent strains or disease-causing strains of bacteria very quickly from those that are usually non-pathogenic. Virulent, again, is disease-causing. Remember, that was Griffith's work. He witnessed horizontal gene transfer through transformation. This is how that E. coli, when we hear about the good and the bad E. coli, this is how we happen. We think that probably there was a phage DNA, that was the vector that actually moved genes between these strains of E. coli and caused some of those pathogenic strains of E. coli to come out. Now, we often find that one of the characteristics that we see in those pathogenic strains are these adhesive fimbriae that I introduced you to. So that's another way that when we're classifying organisms, trying to figure out who they are and could they be the problem, is we look for these adhesive fimbriae. Typically, we don't find those in non-pathogenic strains, but the ones that cause disease, they tend to be, because again, for adherence, right? And stick to things like your tonsil cells. Okay, that's it for the lecture. I'm going to stop the recording.